So next up is information on managing white mold in soybean. And that was a fairly common occurrence last year. This is coming into soybean. And so we have Dr. Michael Wunsch to discuss the subject. Michael is originally from Montana. He got his PhD from Cornell. And he's been here at the center since 2010. So he's gone through his fifth season already. And Michael works with a lot of different crops, but primarily pulse crops, and then also soybean and dry bean. And white mold must be his favorite disease. He spends a lot of time with it. So anyway, without further ado, I'm going to let Michael visit with you and share some of his research and his recommendations to keep it a minor disease in the future.
As Greg mentioned, we have a very active sclerotinia disease management program here, research program, targeting primarily dry bean, sunflower, and then also soybeans. Okay, basically the philosophy that I'm following at this point is we're doing intensive work right now on sclerotinia. It's less of an issue in this region as it is as you go further east into the Red River Valley. But we're doing intensive work in sclerotinia right now, and once we have addressed some of the critical questions that remain unanswered at this point, then I intend to transition, and we're going to look probably quite intensively at some of the root diseases. Okay. But right now we're doing a lot of sclerotinia research, and this talk will not focus that much on our own research, although that does come into play as we move on here. Okay, so sclerotinia management of soybeans. Now, irrigated soybeans, sclerotinia is of course a recurrent and often very economically important disease. Dryland soybeans, of course, this is a sporadic disease, often quite modest in its implications relative to yield, except for low-lying spots, okay, wet spots in the field. Now, I will be giving this talk targeting the dryland grower, okay? Certainly all information presented here is relevant to irrigated soybeans, but I am really targeting a dryland producer in this context. And in irrigated soybeans, we'll be talking a little bit more about water management. Okay, so this is what we will talk about today. And the first question that I want to talk about, and we're going to spend probably half the time on this question, is when do we need to worry about sclerotinia? Like what are the factors that make a year a high-risk year for sclerotinia? Okay, and it's more than just cool temperatures and rainfall. Okay, it's more than just that. And so we're going to talk about that in detail. We'll be pulling in research conducted across the globe, actually, to help you get a better understanding of the environmental conditions that favor sclerotinia and make a particular year a high or low-risk year for the disease. Then we'll look at three areas of sclerotinia management, focusing on those three that I think are probably highest on people's radar screens. And the very first of those things, of course, will be row spacing, which I think is the first thing that a dryland producer thinks about doing to avoid future problems. Okay, so we're going to talk right now about how do we assess the risk of sclerotinia. And to do that, we're going to review the sclerotinia life cycle very briefly. Here I've got some sclerotia that I put on a rock. Okay, those are the resting structures of the fungus produced in diseased tissues. They fall into the ground, into the soil, of course, and they can persist in the soil for many years, as you well know. Now, when those sclerotia are in the top 3 inches of the soil, but particularly the top inch and a half, they can germinate if conditions are favorable to form these little mushrooms called apothecia, and they are little. I mean, the largest ones I've ever seen approach maybe a ½ inch in diameter, but usually you're looking at about a ¼ inch in diameter, even an 1/8 inch in diameter. When the spores produced by those little mushrooms are released into the soybean canopy, their primary place where they actually colonize and cause disease would be the dead blossoms. Basically, this fungus is unique in that it does not have the ability to land onto healthy tissue. The spores cannot land onto healthy tissue, colonize that healthy tissue. It must land onto dead tissue and basically get a running start in that dead tissue. Okay, and then you get your disease. Notice here that we have the disease originating okay, where pods are, where you originally had a dead blossom. And but what's also very critical for Sclerotinia, and this factors into disease management and plant populations and row spacing that Greg was mentioning about an hour ago, is you also have secondary cycle disease caused by plant-to-plant spread. Okay, this is not like other diseases where you have spore production that causes secondary cycles. You do not have any spore production on these plants. But what happens is if you have a diseased plant in contact with a healthy plant, that the fungus can move from the diseased tissue into the healthy tissue in the neighboring plant. Okay, and then you deposit your sclerotia in the soil. Okay, so first off, we're going to talk about this step right here. You aren't going to get spores unless you have little mushrooms. Okay, so we're going to talk about what conditions are favorable for the mushroom production. Okay, and these are mushrooms, and so they like soil to be wet. Okay, this is a laboratory study, but it's very nice and illustrative. Basically, what you want to think of are kilopascals are measures of soil moisture. The more negative it is, the drier it is. Okay, closer to zero, the moister it is. And what they did here is they had sclerotia in petri dishes, and they either added water when the soil didn't really dry at all, i.e. they kept the soil moist the entire time, that's this treatment. Or they let the soil dry to different levels before they rehydrated. And they did this for an extended period, and what you can see there is that fluctuations in soil moisture cause sharp reductions in the percentage of the sclerotia that germinate to form these mushrooms, and very sharp reductions in the apothecia production. Okay, and now this is the lab study. Now I'm going to show you data from the field that support this exact same trend. So this is a field study, microplots in this case, conducted in Sweden. Okay, I said data from around the globe. Okay, and in this case they applied 0.08 inches of irrigation every day, and they excluded rainfall. Okay, and what you can see, they buried the sclerotia right at planting. Okay, and then they assessed when they got apothecia production and how many apothecia they got, and you can see they got apothecia production within 24 days of planting and burying the sclerotia. Okay, I didn't even have a close canopy there by any means. Okay, but they kept that soil moist the entire time, favoring apothecia production. Now, they applied the exact same amount of water, but now 0.24 inches every 3 days. And what do you get there? You get drying of the soil between irrigation events. And you can see that we didn't get very much of an initial delay, but there certainly is an overall delay in apothecia production and an overall reduction initially. So the next step, which is to apply the exact same amount of water, but now 0.56 inches every week. Okay, same amount of water distributed differently. Now you really get some soil drying between your shots of water, okay? And here, now you have a delay of over 2 weeks in the production of the first apothecia, and you have much lower apothecia numbers. Okay, so right here, you can see right here is that if you have sustained soil moisture, then you're at risk for apothecia production. Okay, if you have soil fluctuation, a fluctuation in soil moisture levels, particularly extended dry spells, your risk of apothecia production is much lower. Okay, now this translates also into your soil type. Okay, because soil types, of course, some soils retain water better than others, right? And so here they have they tracked the production of apothecia in sandy soils. Okay, and you got your first apothecia production 32 days after, again, this is the very frequent irrigation, okay, 32 days after burning the sclerotia in the sandy soil. Now you take a loamy sand, has more organic material, can retain water a whole lot better, and you get a lot more apothecia production. Okay, so what this means is if you have heavy clay-based soils, you're inherently at a higher risk of apothecia production than if you have a well-drained soil. Okay, and you've just got a higher risk environment for sclerotinia. Okay, so what do we worry about when we think about soil moisture? What's the depth we care about? We only care about the depth from which these sclerotia can germinate to form the apothecia. Okay, so if you have sustained soil moisture 6 inches deep, that doesn't matter for sclerotinia. What matters here is the first 2 to 3 inches of depth. And unfortunately, we have no local data, but here's some data from the UK, from California, and Israel, okay, that all point to the exact same story. that this particular fungus, Sclerotinia sclerotiarum, can only germinate from about 2 to 3 inches deep. And my personal observation in the field out here, I haven't dug and run numbers, if you will, but my personal observation, I mean, I would look for a lot of epithecium, count a lot of epithecium in trials, is that this holds about true for us. I would be astounded. I don't think I've ever seen anything germinate 3 inches deep. Okay, 1 to 2 inches is typical. I'd say 1 inch is most typical, half inch to an inch. Okay, and so what we care about are the top couple inches of the soil. Okay, now it's not just soil moisture though, soil temperature too. This is a cool-loving fungus, and it likes soil temperatures cool. And so here you have a laboratory study. It'd be a little bit hard to run in the field. And they used different individuals of the fungus and grew them in the laboratory in saturated soil at different temperatures. And you got virtually, like got 90–95%, virtually 100% success in terms of germination of those sclerotia under temperatures of about 59 Fahrenheit. You raise it up to 68 degrees Fahrenheit and you can see a reduction. And I would anticipate that that will reduce even more as you get warmer soil temps. Okay, so cool soils, cool moist soils. It is a mushroom, so that makes sense, right? Now, the next thing is cool and moderate air temperatures. This is no surprise. Here's a laboratory study just showing the growth of the fungus on petri dishes at different temperatures. And what you can see is that the ability to grow drops off precipitously around 85 to 95 degrees Fahrenheit. Okay, and a parallel study here done in the greenhouse, inoculating dry beans in this case, we don't have soybean data for this, shows the rate. So this is hours after inoculation, okay, and this would be the Sclerotinia severity, the rate of disease progression at different temperatures. And what you can see is that 50 degrees Fahrenheit, the disease progresses relatively slowly. As you increase your temperature to about 77 here, we have very rapid disease progress. But look at this, 86 degrees Fahrenheit. You have virtually no disease development in this case. Now this was done with one fungal individual. There is variability in the fungus. And so I would expect that some fungal individuals will be able to cause more disease at 86 degrees Fahrenheit. Okay, but it is illustrative that this is a cool weather disease. These numbers actually correspond nicely with this. Okay, and what your real risk factor is are temperatures in the 60s, 70s, and perhaps lower 80s. As you get into the 90s, your risk factor drops a lot. What's interesting here, when they ran this study too, what they found, there was another parallel study they did here. What they found is that if they kept the plants at 77 degrees Fahrenheit for a period, and then subjected them for a day at 86, and then brought it back to 77, that day at 86 significantly cut down the rate of disease progression. So basically, even temperature fluctuations that include really high temperatures are going to cut down on your disease progression. We don't have really good data, but the data that we do have, which is kind of observational and anecdotal, suggests that you need 40+ hours of sustained wetness on the plant surface to facilitate that initial infection event and development of disease. And so what this means is that you would need sustained high relative humidity in the canopy. And this is based on data collected in the field, watching the progression of Sclerotinia epidemics in soybeans relative to environmental conditions. And so I say under typical field conditions, this means you have very variable temperatures. Okay, but there is evidence that when you have really cool temperatures, you don't necessarily need that much humidity. Okay, and this is really interesting. And so here, these would be greenhouse studies here, and in this case, we've kept the temperatures constant at either 64 degrees Fahrenheit or 72 degrees Fahrenheit, and you can see you had 90% relative humidity or 25% relative humidity, and they inoculated just like it would happen in the field. They took a dead blossom, They applied spores of the fungus to that dead blossom, and then they put that dead blossom into the lowest node of the plant. So it's just like you'd have in the field, and they kept those plants either at 90% or 25% relative humidity. Okay, and what you can see is that even at this low relative humidity, they got a fair amount of disease. The disease development was certainly dropped off as your temperatures rose. Okay, so the ability for Sclerotinia to establish itself under lower levels of relative humidity, actually really quite low levels, are probably limited to the coolest temperatures. But if you have an extended period of temperatures in the upper 60s, low 70s, even mid 70s, you should consider yourself, you should be, and you have the soil moisture to favor the production of the apothecia, you should be cautious because you don't necessarily need a really sustained wet plant surface to get infection under those conditions. Assessing risk of Sclerotinia, sustained soil moisture, cool soil temperatures, cool air temperatures, and generally high relative humidity, although that might be less important if temperatures are really cool. Now, what I want to talk about with the soil moisture that I didn't mention is that the rule of thumb is that to get the apothecia, you need about 2 weeks, approximately 10 to 14 days of sustained soil moisture in those top few inches. Typically what happens is that the apothecia start germinating earlier in the growing season, okay, and then you can get some dry spells and their development just freezes. But then if you get about 2 weeks of sustained soil moisture in those top couple inches, say after the canopy closes or as the canopy starts to close and protects that ground, in that period, that's all it takes for the sclerotia then to reinitiate that growth process and produces apothecia. So yeah, 10 to 14 days during, leading up to or during the bloom period of sustained soil moisture is your risk factor. Okay, so we're going to talk about row spacing. Okay, this is the first management strategy that we'll talk about, and these are the results you would expect, right? Sometimes you can increase your yield, okay? Look at this though, these are data from Wisconsin from the late 70s, and you can see 15 and 30-inch rows, various soybean varieties. I want to point out to you a couple things. You had astronomically higher levels of disease in the narrow rows than the wide rows, okay? And your seeding rate was extraordinarily high in the narrow rows, okay? And what my interpretation of this data would be is that you had really favorable conditions for Sclerotinia leading into bloom and in that first period of bloom. And then it dried out. And so basically, the 30-inch rows were probably completely wide open at that point, or pretty much wide open. And you had a closed canopy in your 15s. But this is again with that plant population difference. Again, with this same plant population trend or seeding rate trend, here's some more data. This is from Ontario. And here you see that same disease response, more disease in the narrow rows, as you'd expect, but that response is much more modest. Okay, and you'll see now that you no longer get the yield response that you want. Okay, we all know that there is a yield penalty to planting in wider rows, and so basically, if you don't have enough disease pressure, you're not going to make up for that yield penalty, is what this is saying. But what's really, really fascinating are these data from Michigan. Again, those studies all increased the plant seeding rate as you narrowed up the rows. Here what they did is they tested that. Okay, you increased your seeding rate as you narrow up the rows, but why don't we test the same seeding rate too? Okay, and so they have, this is actually, and there's your seeding rate, and what you can see right there is that when you tested at keeping the seeding rate constant, i.e. you drop your seeding rate in those narrow rows from what you were testing before, or planting before. I don't know if that was the recommendation in previous time. But what you can see is that you suddenly drop your disease, you increase your yield. So why would you drop your disease? Remember, when I talked about the disease development, I said that you can get infection from the spores when they land on the dead flowers. And then what you can also get when you have a diseased plant in contact with a healthy plant, you can get plant-to-plant spread of the disease. Okay? When you have the narrow row spacings, you have increased your spacing between individual plants. Okay? In this case, Your plants, in this case, the distance between individual plants within a row should be should be 4 times as much as this. Okay? So in the 30-inch rows, you have within the row plants in very close succession, i.e. they're touching each other. Okay, and so you have lots of opportunity for that secondary spread of sclerotinia. Lots of opportunity, and the limited data that we have suggests that actually plants are more susceptible to that secondary spread. They don't require conditions that are as favorable for sclerotinia to get that secondary spread as you require for that initial infection. So secondary spread is really a serious concern, and I know I have seen it anecdotally where you get these patches where I've seen this in fields planted with a large planter when you hit different conditions, and you get really dense patch in a stretch. And in that stretch, you get a lot more sclerotinia because you have a lot of plant-to-plant spread. Okay, so here are some of our data here. Again, keeping the seeding rate constant. We're seeding at 165,000 pure live seeds an acre for a target plant population of 150,000. Okay, and these are data where we use irrigation to facilitate early, intermediate, or late disease onset. And what you can see right here is that we consistently get higher disease in the narrower rows, but you always have higher yields. Here are data from evaluating this exact same question on 14 different varieties. Conducted last year. We conducted this again this year. We don't have those data tallied yet, but still processing seed yield and quality. But what I want you to look at right here is you almost have a bell curve effect where you have the highest disease levels in the intermediate rows, and the very lowest disease levels, of course, on the widest rows. But the second lowest are typically the 7-inch rows. And what this tells you, again, you've got this tension here. The wide rows give you increased airflow to reduce initial infection events, but they also have really dense planting within the rows, so you have a lot of plant-to-plant spread. Okay, in the 7-inch rows, in that solid-seeded soybean, you're going to have probably more initial infection events, but very few opportunities for secondary spread, because the plants are spaced further apart. We're definitely dropping off yields, almost universally. I think actually universally on those widest rows. Every one of those varieties. Okay, and it looks like it's actually a wash between the 7-inch and 21-inch rows in terms of yield. Okay, and disease levels are actually very comparable, all things considered. Definitely at 14 and 21 are virtually done. Okay, so with that, we'll move into partially resistant varieties. You can see, obviously, there are some major differences in susceptibility of varieties, very significant differences, and those translate into significant yield differences where sclerotinia is important. Okay, now, resistance to sclerotinia is not like resistance to rust. Okay, it's not confirmed by a single gene that confers complete immunity. We don't have that for sclerotinia. Okay, so what you have is you have a combination of small genes that all contribute a little bit towards disease resistance. Okay, and that's true genetic resistance of sorts. Okay, it's basically you have a better immune system. Okay, and then you also have something we call disease escape. So these are plant architecture traits that create an environment that is less favorable for disease. And critical ones here are things like plant canopy height. The higher the canopy, the more likely that you're not going to get a whole lot of airflow down through the bottom. Okay, so shorter soybeans tend to develop less sclerotinia. Okay, and then this is critical, the timing of canopy closure relative to bloom initiation. If you have a soybean variety that closes the canopy, say, a week before bloom, another one closes the canopy a week after bloom, in a given row spacing, the one that closes the canopy later, of course, is going to have less sclerotinia. Now, are you going to have less yield potential, as Greg mentioned, overall, in absence of disease? You may. Okay, probably you will. So there is a trade-off there. But anyhow, those are two examples. There are other factors as well. But basically, what I want to stress, though, is that this is not necessarily a silver bullet here. Okay, research from South Africa indicates that the stability of partial resistance to sclerotinia in soybean varieties basically the performance of these varieties in terms of resistance to sclerotinia can be variable across environments. So a variety that's highly resistant in one environment may not be in another environment. Okay, we haven't validated that. It's been an observation from a team from South Africa. No one's validated that. And then our other liability is here, how do we know what variety is resistant? And so a lot of the breeders do provide resistance ratings for white mold, and how good are those? So we have a study this season here where we subjected those same 14 varieties to different irrigation regimes in the critical 3 weeks after canopy closure. So we have moderate differences in environment there. In this case, what you can see is that the performance of these varieties was roughly consistent across those environments. We're going to do additional work in the future here, manipulating planting dates and testing over different locations to more rigorously assess the replicability of resistance results within our environment and soybeans adapted to this area. Okay, but what I want to point out to you is these are the resistance ratings assigned by the companies. And in all cases, I have pairs of varieties from the same breeder and having very similar maturity and differing sharply in the resistance ratings assigned by the companies. So we really looked for varieties that had big gaps in their white mold ratings. Okay, and so if you look through here, you'll see that many of these do hold through. You have Pioneer 90Y21 versus 90Y20. The resistant one clearly outperformed the susceptible. Okay, and you have Proceed 1020 versus Proceed 2030. Resistant clearly outperformed. Okay, and now we have Pioneer 90Y90 versus 90M80, and again you see that exact same reaction. So in this case, our resistance ratings are performing well. As we move down, though, you'll see that some of these don't perform according to the ratings. This Kruger 0402 susceptible consistently performs better than 0504, and that was true the previous year as well. And in other cases, we don't see much of a difference at all. These mycogen varieties are very similar in their performance, even though they're rated differently. And these Kruger varieties, again, same deal. And I'd even say that Pioneer 90Y50 versus 90Y51 is, you can see that rating is not the most important. So basically the take home is the ratings that the companies are providing us are perhaps sometimes, basically they're sometimes useful. Okay, and this is what these initial data suggest. With that, I will not go into fungicides today. Okay, I think that within the dryland producers, that those are the management tools that most people will use. What I'd like to stress, though, is that within a month or so, I'll have fungicide results posted online. You can go to the Carrington Research Extension Center website, click on plant pathology, and click on soybeans, click on sclerotinia, and you'll have the fungicide efficacy results already posted, but I also have timing information on when you should apply fungicides to optimize your performance if you decide to go that route.